So today I would like to discuss with you a little bit about translational toxicology. We do a lot of work in vitro. We do work in vivo in animals, and eventually want to also predict what's going on in humans. Right, so I would like to discuss with you some items in that translation that we are doing. First, a little bit about myself. I'm an associate professor at Maastricht University. I work at the Department of Pharmacology and Toxicology. I was trained as a genetic toxicologist. Okay, so I want to start actually today with my take-home messages, right? So usually I have my conclusions at the end, but today I would like to start with take-home messages because it's a little bit of the flow that I will use during my presentation. I will start with in vitro, then I will go to in vivo in animals, and eventually I would like to go to the human situation. So the take-home message actually is that in vitro tells you what's possible, in vivo in animals tells you what's plausible, but actually if you measure in humans, that tells you what's actually going on, right? So, but each of these steps has its advantages and disadvantages. For instance, in vitro, okay, it does not tell you what's really going on in a human body, but you can manipulate it more easily, right? You can do it more quickly, less ethical questions, right? So in humans, it's much more difficult to do studies in a real life environment, right? So each site has its each step in this whole translation from in vitro to in vivo has its advantages and disadvantages. So just to stay to be on the same page, some definitions. In vitro means within the glass, right? So in your TC flask, etc., which means that you also include cells in there. I've read papers written by chemists, in which they say that if they measure it in cells, it's in vivo, right? So for them, it's a completely different step. But I think for us biologists, I don't have to go very deep in this definition, right? So in vitro is in the flask, right? So that's what we also with your cells. In vivo is when we go to the human situation. We also talk about ex vivo, for instance. That would be if you, for instance, expose an animal to a drug, and then you do experiments with cells isolated from that animal later on in the flask, right? So there are combinations in there as well. By the way, if you have any questions, please don't, please interrupt me. Just raise your hand or ask your question if you wish. And I would like to give you an example of where we think that in vitro is telling us something important, but then in vivo it shows that there's nothing going on. We did a study at Maastricht University in which we wanted to investigate the vasoconstriction, the effect of vasoconstriction by quercetin. Quercetin is a compound you can buy in the drugstore, and it can reverse vasoconstriction actually. So we have an in vitro model in which we isolate the blood vessels from placenta, and then when you if you incubate them in vitro, and you can then look at the concentration, you add some concentration of phenylephrine, which is an adrenaline-like compound, you see constriction. So the vessels constrict. If you add a drug, tamsulosin, right? That's a drug which is a blood-lowering drug. You see that you need higher concentration of your adrenaline to reach the same contraction of your vessel, right? So that's the whole effect of this tamsulosin, which is a blood pressure lowering drug. So because it makes sure that you relax your vessels. The idea was that quercetin could do the same. And so if you are an elderly individual, if you then have that drug, because that drug is usually prescribed to elderly, if you have that drug and you also have quercetin was especially used by elderly men because it was mentioned to be very beneficial for the prostate, right? Then there was the people were afraid, the government was afraid that it could lead to what we call orthostatic hypotension, which means that your blood lowering effect is so strong that if you get up very quickly, the blood in your brain disappears and you faint, right? And then you fall with all kinds of difficulties there. So what happens if we add quercetin to this equation, right? We did this incubation again. Quercetin is actually metabolized to the quercetin glucuronide. This one is usually excreted in the urine, but we see a clear effect of the glucuronide. So we see that in the green line here, you need much higher concentrations of your phenylephrine to get a contraction. So that means that having tamsulosin and having also the quercetin in there, that leads to a severe relaxation of the vessel, and that could lead to this orthostatic hypotension. This is all in vitro, right? So the question was, is that actually also happening in vivo then? So then we went to the animals. We had two types of animals: the rat, the Ricky rat, that's a normal rat, and the spontaneous hypertensive rat. And we measured the blood pressure, which is on the first two graphs, and the heart rate on the next two graphs, and nothing happened at all. So the drug, the tamsulosin, did work, right? Because we see blood pressures going down, especially in the spontaneous hypertensive rat. Heart rate goes up, which is a direct effect of this blood pressure lowering effect. But there was absolutely no difference between the rats with or without the quercetin. Right, so in vitro, we thought, wow, that's a strong change. Right? It was really in orders of magnitude. But then in vivo, we didn't see anything happening at all. Right? So that's already showing you that translating something from in vitro to the in vivo situation is very difficult. And then you can ask yourself, okay, this is a rat. What happens in humans? That would be a different story. This was already shown to you by Sabine, this graph. But I think it's also important to take into account that when you look at this in vitro, in vivo, in human, and in vivo in humans, in vivo in animals, and in vivo in humans, that you are looking at different points in the whole spectrum of toxicokinetics and toxicodynamics, right? So if you would expose cells in vitro, you're actually more looking into the dynamics part, right? Because then you want to see what is actually going on in those cells, whereas the toxicokinetics, you actually then you skip more or less, right? Because you are exposed to a certain dose, it has to be distributed, has to be taken up. If you just add your chemical to the cells, then the whole toxicokinetics is skipped more or less. So you make it more simple, and therefore the translation is not always necessary, or not always very clear, not very possible. So of course, then you can go to animals again to mimic this toxicokinetics. But what we also have is PBPK, which is modeling, right? So then you can have a look at what do we already know about certain compounds, how do they distribute over the body. So what you can do then, so PBPK stands for physiologically based pharmacokinetic modeling, right? So you have your exposure, you know, and then you can calculate how much is taken up, how much is distributed to certain organs, how much in time, right? So you can also follow all the process in time, and you can do that. Let's say for animals, then you can compare that to human situation, right? So that's already important already to see whether a certain animal model could predict what is going on in humans, and eventually there is a large. Oh, that was the wrong button. Well, I can go back like this. Yes. And eventually, of course, you have your population. There's a large variation in the population. I will talk about that on Thursday when we talk about susceptibility. And you can even model that in these PBPK models, right? So you take the toxicokinetics, put that in this is all in silico, right? You can always calculate it. And that is based on some physiological properties of the compound that you investigate. So then you go to the in vitro model. I think most of us use simple 2D models, right? So you have your cells in a flask. Who's doing in vitro work? Maybe you can raise your hand. Who is doing in vitro work with more complex models than just 2D? Nobody. Okay. Yeah, that's what I expected, right? Because most of the time we start with the very simple models, just 2D, your mono layer of cells. You put your cells on there or your compounds on there, and you want to see what happens. But I think there are quite some opportunities also to change it, to improve these models. You could, for instance, improve the monoculture itself, for instance, by introducing day-night rhythm, right? So you can sync, or usually in your cells are not what we call synchronized, but you can make them synchronized, and then you can study things over time, changes over time. We can do co-cultures. We do a lot of co-cultures in Maastricht, in which we, for instance, want to mimic inflammation. So we have the monoculture of, for instance, epithelial cells, and we put on top, inflammatory cells from different origin. And then either directly we mix them together, or we work with inserts so that you have 2 layers of cells. So there are different possibilities there. We can work with spheroids. So usually we work with the monolayer, which is a flat cell, but in real life, cells are not that flattened, right? So then you can better maybe work with spheroids. You can even have spheroids with mixed cell types. We used in the past a spheroid mix cell type for brain. So you make sure that all cell types are in there. And of course, you can also take a piece of tissue from an animal or a human, and you can put that into culture. Right, so there are all kinds of possibilities here to improve your in vitro model to make sure that it mimics the in vivo situation to a better degree, to increase the extrapolation to the real life in vivo. And there are even further developments. Again, I'm not the expert in this one, so I don't want to go in too deep. I just want to say that you can either have interaction, like this is a model for the lung. You have airflow and blood flow with your cells in between, and with the vacuum on the left and the right, you can even mimic the breathing effect, right? So that your cells are stretched and relaxed, etc. And I know, for instance, that in Health Canada, in Canada, at the institute, the health institute there, they are working with this kind of with the microfluidics. So what they have cells of the lung and cells of the liver, and what you can actually mimic then is metabolism in the liver, and the metabolites are transported via the microfluidics to the lung to see what the effects are in this combination. Right? So you can make it as complex as you want. So that's the in vitro situation. So then, of course, we would like to go to animal models, and then you would like to ask the question, okay, how does that translate to humans? And usually what you can do is what we usually do is we take mice or rats, because these are available. But sometimes you can wonder, is a mouse or a rat the most suitable model? And maybe that is something that we should think about more seriously, right, before we just start with mice or rats. I just have an example here again. If you would like to study the PTH1 receptor, so parat hormone receptor, if you're interested in version 1, so gene 1, you can either use a pig, because genetically this gene is much closer to the pig than to any other animals. If you are interested in the parat hormone receptor 2, you might use the rat to study that. Right? Usually, and they are in the same pathway, right? So then you could wonder, okay, should I now use a rat or should I use a pig? Who knows, right? So what is the best model here to choose? Another example is up here. It's the guinea pig. So this one is, for instance, in the transporter, the ABC transporter B1. So if the human is very closely related genetically to the guinea pig. So maybe the guinea pig, if you study that compound, that gene, or a compound that affects that gene, then maybe the guinea pig is a better model than a rat or a mouse. So an example here. Again, we studied atherosclerosis some time ago, and we wanted to see how chemicals affect atherosclerosis. And if you do that in mice, you cannot do that. You can feed a mouse a high-fat diet, it will not develop atherosclerosis. You can feed them a lot of fat, and they will not develop atherosclerosis. So what has been done over the years is that they developed knockout models, right? So a knockout of the ApoE gene or a knockout of the LDL receptor, and these animals, they develop atherosclerosis, even spontaneous, without a high-fat diet. And if you give them a high-fat diet, it becomes more severe. But this is a very extreme situation, right? So even without the high-fat diet, these animals already develop atherosclerosis. So the problem that we encountered is, so how can we see an effect of our chemical on whether it accelerates the atherosclerosis if the animal itself is already developing atherosclerosis so quickly, right? So maybe we are not looking at the right model. Anyway, we did the studies in these mice, and I think what we expected or what we were afraid of actually happened, because we didn't see a lot of things happening, and because we think that's because the model was too strict, too strong. What animal could we choose here? What would have been better? What animals develop atherosclerosis next to humans? You probably don't know. Chicken, right? So chickens, they develop atherosclerosis just like humans on a high-fat diet. So maybe we should have used chickens, right? And there you also have the possibility maybe to go into in vitro work, because we have eggs available and we can work with that as well. Right? So sometimes you have to choose another model, but because chicken are much more difficult to keep in a lab, we often switch to those knockout models and mice and rats, but that's not necessarily the best choice. Keep that in mind. So then we have in vitro in human cells, we have in vitro models in animal cells, we have in vivo in rats, and we want to predict what's going on in the humans, right? So we make this parallelogram, as we call it, right? So if we know what is the difference in vitro between the human and the rat, for instance, and if we then have the rat also in vivo experiments, then maybe we can predict what is going on in humans. And if we go to high throughput data, that might be a good way to do a good prediction, right? So we have a lot of data there, and we can maybe predict, but it is quite challenging. And of course, it's logical to take a human or to take a mouse or a rat to predict what is going on in humans, because there are quite a lot of pathways and systems that are very similar when we look at mice and humans. Some examples: immune system has overlap with the immune system in cardiovascular disease, in this case of humans, and you can see a whole list of pathways that are very similar or have overlap with the human situation. But there are also, of course, systems that have no overlap at all, that are either only present in the mouse or only present in the human, right? And then you cannot have a look at it anymore, or you cannot use the mouse anymore, right? So be aware of what is there in the mouse or and whether it reflects what's going on in the human. Also, with regard to the immune system, this list actually says that there is an overlap between the immune system in the mouse and an immune system in the human. Of course, it looks very similar, but there are some significant differences. I just put some small differences here already on the board. Neutrophils in the peripheral blood humans, we have 50 to 70% of our white blood cells in the circulation are neutrophils. In the mouse, it's only 10 to 25%. In mouse, the majority of cells are lymphocytes. In the humans, it's not, right? So the blood composition in the mouse is already different as compared to the human. Expression of CD33, right? So these immune cells, they express these CD molecules on their surface. In the human, it's expressed in the monocytes. In the mouse, it's expressed on the granulocytes. So it's a different cell type, right? So there are clearly differences between those two cell animals, right? Humans are also animals. CD4 on macrophages, absent in the mouse, present in humans. So if you study CD4, you cannot use the mouse. IL-13 effect on B cells. This one is not non-existing in the mouse, but it induces a switch to IgE in the humans. And the list goes on and on, right? So overall, there is an overlap in the system. But if you go to look into details, if you want to study certain details, you may not be able to use a mouse at all. And the next thing is something that I have to mention, because I did that myself. I tried to isolate granulocytes from red blood, because that was part of my PhD. I wanted to add an experiment in my head during my PhD. And I never succeeded because rats don't have granulocytes. That's not true. They do have granulocytes, but only if you induce an inflammation. Healthy rats, and I was using blood from healthy rats, they don't have granulocytes in their circulation. So I worked for 2-3 months to isolate those cells, which simply did not exist. So also take account on realize what the physiology is of those animals that you use. So if there is quite so there is overlap, but if there are important differences that you want to take into account, I think it's important to look for other models. For instance, pig. We used piglets, right, in one of our studies, but that is of course much more difficult. These animals are bigger, and therefore there's a much more costs involved and much more work involved, and so on. Right, so a good model is not always the best model, and the best model is not always good to use. So if the mouse is not mimicking the human good enough, maybe we should make the mouse more human-like, and that is something that has been done in the past, right? So we use knockout or knock-in models. So we add human genes into a mouse to see what is going on there. So that's what we then call humanized models, right? So we switch off more or less the mouse gene and we add the humanized gene in there, the human gene in there. One example for that is the p53 mouse, right? So what we did there, or not we, what has been done there is that you knock out the animal form of p53 and you add the human form of p53 to the animal, right? So as to see also what the effect is of human p53. An example that I want to give with regard to making a mouse a better model for humans is with beta-carotene metabolism. There were 2 studies, the ATBC study and the CARROT study, that studied whether smokers would benefit from vitamin A. So these smokers got extra beta-carotene. The reason was smokers have a lower beta-carotene status. There is increased oxidative stress, and vitamin A is an antioxidant. So there's more oxidative DNA damage. So let's intervene with oral beta-carotene. So these were huge intervention studies done in Europe and the United States. And what eventually turned out is, so this is mortality, and this is the lung cancer incidence. What you see is that those who got the beta-carotene had a higher mortality and a higher lung cancer incidence. So it did actually the opposite of what we expected. So these two cases, these 2 trials were stopped immediately with the, of course, when they did an intermediate analysis, then they found this inverse effect, and then these were stopped immediately. Well, that's, of course, something that's not nice. So we wanted to see, okay, what's going on there. We wanted to use a mouse model, but the mouse has a problem, because if we give beta-carotene to a mouse, it converts the beta-carotene to 100% to retinol. And we humans, we only convert about 70% of our beta-carotene to retinol. And as a result of that, beta-carotene cannot accumulate in mice, but it can in humans, right? So one of the enzymes that is central in that is the enzyme BCMO. So what we did is we had access to a BCMO knockout mouse, and we thought maybe this is a better model. And when we look at indeed accumulation of beta-carotene in serum and accumulation of beta-carotene in the lung, then we see that indeed in the knockout model, so in the knockout mouse, we see an increase of beta-carotene in both the organ and in the plasma. Right? So we thought, okay, the BCMO model would be a better mouse model to mimic the human situation. Right? So this would be a humanized model. And then actually it didn't work as well, because we thought it would be a good model, but it didn't work, because if you knock out the BCMO, the mouse does not convert your beta-carotene to vitamin A, and what we saw was actually a vitamin A deficiency in those mice. So all the effects that we saw were actually a vitamin A deficiency and not accumulation of beta-carotene. Right? So this is really something that is really, so we were very lucky that we had quite a lot of controls built in, and as a result of that, we were able to find out what was that this model actually was not suitable anyway. These animals, because of the vitamin A deficiency, it led to a large amount of inflammation in the lungs, and you can see that here, right? So these patches of inflammatory cells in the lung were completely absent in the BCMO positive animals. So this was an extreme situation, and it was not mimicking the human situation. It was actually mimicking the human situation more than the original mouse, but because you changed the mouse physiology so much, it was not working anymore, because we were inducing other side effects, which was the result of the vitamin A deficiency. So was the whole study for nothing? I don't think so. We still got quite some good insights in lung carcinogenesis, and what we actually found was that there's a major role of what we call the pulmonary neuroendocrine cells, the PNECs, in especially the small cell lung carcinoma, right? So the model itself did give us some nice clues anyway. So we saw something happening in the humans. We tried to mimic it in the in vivo situation. That didn't work, right? So because we got the wrong or too strong mouse model. So then we thought, okay, let's then go back to the in vitro situation and then see what's going on. And we know that smokers have a lot of inflammation. And what the hypothesis was is that so during inflammation, you make a lot of radicals, which are usually, one of them is hydrogen peroxide. Hydrogen peroxide is actually converted into water by catalase, but it can also be used by MPO, myeloperoxidase. And what we saw was that if we add the beta-carotene metabolites, then we block this gene, which means that we have more hydrogen peroxide, and as a result of that, we have an increased Fenton reaction, leading to more hydroxyl radicals, and therefore more oxidized DNA. And that's actually what we showed with the COMET assay, right? So you know the COMET assay? Yeah, I see people nodding. So this is the FPG COMET assay. What we see is if we give vitamin A retinol or retinoic acid, right? So these are the vitamin A metabolites. We actually see an increase if we have that combined model of inflammatory cells and epithelial cells. We have an increased DNA damage, right? So that's actually one of the reasons why we thought, okay, this is a possible explanation. So on one hand, you want to go from in vitro to in vivo to understand, but in this case, we actually went from in vivo back to in vitro to know the underlying, potential underlying mechanisms. So eventually, you would like to go to humans, because then you don't need to extrapolate, right? But there is a problem with humans in your studies, because mice, they are genetically identical, so each mouse is the same. Whereas we humans, we are genetically diverse, and you also have less control over the environment that people live in, right? And a lot of our traits that we have, they are, let's say, continuous traits, right? So it's not that you are, if you look, for instance, for skin color, you're not black or white. There's everything in between, right? And this is so if you look at, for instance, skin tan in humans, yeah, you can go from very light to very dark, and then you get such a curve. And the reason for that is because most of the traits that we have is what we call polygenic. Not polygenetic, polygenic. That means that more than one gene is involved in the phenotype that we see. Skin color is a very simple example, but you can imagine if you look into pathways, also there, multiple genes are involved, and people may vary between each other in those genes. And that's something I also will discuss in my presentation on Thursday when we talk about susceptibility, right? So there are multiple genes determining your phenotype, right? But it's the variation in those genes that determine the effect. This is a very simple example. If I would have 3 genes, oh, I have to go there. If I would have 3 genes, A, B, and C, if they mix up in the F1 and then I cross the F1 generation, I can get all these combinations of those genes. If I'm going to count just the number of, let's say, black dots and white dots, what you get is something like this. So your genetic variation is already closing into a normal distribution, right? So, and this is an example of 3 genes. If you look, for instance, to the genetic effect on body weight or body measures, all those blue dots that you can see are all genes or genetic variants that add to the variation that we see in body weight and body measures. Right? So if you already have this nice variation with 3 genes, this is hundreds of genes or more than that. So you can see that it's an enormous spread in the population that we have. That would be genetic variation. If we look at environmental variation, so this is a homozygote line of beans, right? So these are genetically identical. If we then take the weight of them and we count the number of individuals, even then we see a normal distribution, and that's because of the environment that's on there, right? So one pea has for instance, or bean has a lot of sunlight, one bean gets a lot of food, etc., and grows larger than the other one, right? So the environment also plays a role, which means that we have these multifactorial traits, and that makes them even more complex. We have the genotype, but we also have the environment playing in there, right? So it's making it more complex. These multifactorial traits are affected by genetic factors, but also by our environment. And if you then look at the total variation, we can look at the total variance is your genetic variation, your environmental variation, and the interaction between those two, right? So then if we try to do an in vivo model with animals, the genetic variation is, let's say, close to 0 because we think they are genetically identical. They are not, but we think they are. At least for a large part, they are genetically identical. So all we look at is then your environmental variance. And what I think is also important, something that we have to take into account, is the parental effect. So if you design your experimental, your animal experiment, I think you have to take into account where the animals come from, right? So you have one father, usually you breed them with multiple mothers, and then you have multiple children. So because the example that I want to give here is the metabolism of benzoyl. It's an environmental pollutant. It's metabolized by cytochrome P450s, 1A1, 1B1. And what we did is we actually did a study in which we we exposed the father to benzoyl, we crossed them with unexposed females, and then we looked at the offspring, and we assessed the expression of cytochrome P450 1A1. This is the maternal ID. And what you see is actually, if you would look at the variance, and each dot is actually 2 siblings from the same nest, 2 brothers or 2 sisters. I don't recall. I thought it was 2 sisters, but I don't know for sure. And what you see is that those brothers and sisters within one nest are usually closer to each other than the brothers and sisters between nests. So the variation between nests is bigger than the variation within a nest. And I think that is something very important for if you do an in vivo study in animals, because you buy the animals usually at a company and you don't get information of whether these are brothers or sisters, or maybe they come from similar nests. You just got these mice, and then you put them randomly, you try to randomize them into the 2 groups. And then you hope that you do not, I would say you hope that you don't get brothers and sisters in one nest and the other brothers and sisters in the other group. Because if you do not randomly allocate them, you can actually have, let's say, what we call a type 1 error. Because if you would have your controls on this side and your exposed ones on this side, then you find a difference simply because they were from the same nest, right? So instead of the real exposure, right? So this randomization is really important, and I think it's also important to know whether these are siblings, yes or no. And it also affects your power analysis, so the amount of animals that you need. If you have more animals from the same nest, that means that you would have a lower power, so you have to use more animals if you have more than one from a nest. So that's usually why we say, when we do our studies, we always take 1 animal per nest to do the analysis. Then to humans, my last part. So eventually you would like to measure in humans, but that's quite difficult, right? Because in humans, you can usually only get easily accessible tissues. Think about blood, right? That's easy. But you cannot get a piece of liver, or you cannot get a piece of lung, although you would like to have, but it's not possible, right? So what we always do is we use biomarkers. And biomarkers are indicators of events in biological systems, and they represent a change in that system because of the exposure, right? So if you have a biomarker of exposure, you want to see a change in that system because of the exposure, right? And it can be on any level. It can be molecular, can be physiological, heart rate, for instance, subcellular, changes in structure, etc., etc. Right? So if you have an exposure and you see same changes, that could be a biomarker that is measurable in any biological media that you can collect from humans. So what we actually want to do here is we have our exposure on one hand, we have the disease on the other hand, and the process that is in between, we want to open that up, because usually that is a closed box, and we want to see what's going on there. And we can do that by, you see, this is actually the same, this is actually inside the box, right? So you can have a look at the internal dose. I think I have it there. For instance, then you measure a compound in urine or in plasma, right? So you can measure, of course, exposure outside, if that's the concentration of a compound in the air, but that does not mean that you know how much got into the animal or into the human, right? So that's why you can measure it in urine or in plasma. You can look at the biologically effective dose. So how much of that compound that enters the body does also actually cause the damage. Then we go to a biological effect. That could be mutations, right? Because DNA damage, not all DNA damage will convert into mutations, right? So that is something that, so you can build that up actually, and then you have susceptibility factors that determine how quickly you go from left to right. Again, these susceptibility factors are something that I will address in more detail later on. If you do biomonitoring in humans, I think it's very important that that a few aspects of your biomarker need to be addressed. First of all, if you want to use the biomarker as a marker of exposure, there needs to be a correlation with exposure, right? So it should go up if you are exposed or not. Usually, if we use white blood cells, but you want to say something about lung or liver, what you hope, of course, is that there is a correlation with your target organ. You also should know the stability and the kinetics, because that determines when you should measure, right? So if for instance, when we look at cotinine levels in smokers, if we ask somebody not to smoke for a week, and we measure cotinine levels in the urine, for instance, it disappears, right? So you need to know at what time do I have to measure to see a certain effect. You have to take into account temporal or seasonal variations. For instance, for DNA damage, we often saw higher levels in the winter than in the summer, because lifestyle is different. People close their windows, people eat differently, etc. But also, if you measure in for instance, red blood cells live 120 days and then they disappear. So all items you measure in a white blood cell, human white blood cell, disappears in 120 days, right? So that's something you have to keep in mind as well. And then you would like to know which host factors affect your biomarker, like age, sex, genetic background, and so on. Okay, so I'm almost at the end of my presentation. Am I in time, Sabine? Not completely. 2 minutes. I'm almost done. So we talked about in silico, right? So that you predict what is going on in a human body by PBPK modeling. We've talked about in vitro models. We can make them more complex to mimic the human situation, and we try to understand what is going on. In vitro models have an advantage that that you can change situations much more easily as compared to in vivo situations in animals and humans, right? And also here, you need to take into account, if you want to make that translation from in vivo animals to in vivo humans, is all kinds of items like, am I looking into the correct animal model? Can I humanize the model? Is the model not too extreme, etc., etc.? So all kinds of knowledge, what you need to make a good translation of knowledge. So as a conclusion, I think in vitro, there are improving models, or models are improving to get better results or better predictions of what is going on in vivo. I think for the in vivo animal studies, you should choose the best species or adapt it to mimic the human situation, or at least try, or try to understand where the differences are. And in vivo, in humans, I think there's a huge variability in populations, and that is also something that we have to keep in mind when we do our experiments. So these were my take-home messages that I started with. So it's my last slide. So in vitro tells you what's possible, in vivo in animals tells you what's plausible, and in vivo in humans tells you what's actually happening or not.